Hi, everyone. I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas San Antonio's neuroscience research podcast. Today is May 6th, 2021, and we're talking with Josh Goldberg, who is Associate Professor of Medical Neurobiology at Hebrew University of Jerusalem. Hi, Josh.
Hi, how are you?
So Josh's lab combines neurophysiology, advanced imaging techniques, and mathematical modeling to understand how the biophysical properties of autonomously active neurons contribute to their degeneration, as well as the pathophysiological adaptations that follow degeneration within neural networks. He uses a variety of preclinical animal models of Parkinson's disease and other movement disorders to model the earliest time points for diagnosis and intervention. Josh is also a Wilson Lab alum and therefore family. So welcome back, Josh. And we've got with us also today, Charlie Wilson. Hi, Charlie.
Hi.
So Josh, you've been working on working on defining the earliest phase of Parkinson's in this long preclinical prodromal period before we see any motor symptoms at all. So it's super interesting to me that you're I guess, I think of you as a biophysicist who spent most of your time thinking about autonomous pacemakers and ion flux. I'm sure there's a lot more to you, but that's how I think about you. But you've worked so long at that scale, yet you're so earnestly working out all the causal chain that leads and connects this scale to human clinical symptomology. So it seems like so much of what we know about Parkinson's is rooted in midbrain dopamine and in pharmacological and transgenic models of Parkinson's. And you're kind of pivoting away from that now. You've done a lot of that, but you're doing something a bit different. Tell us about your angle and how you're getting at this prodromal phase and what that is.
Thanks. So Parkinson's disease is a movement disorder, and we know that the movement disorders, the symptoms, bradykinesia, akinesia, difficulty to move, difficulty, slowness of movement, tremor, are all associated with the loss of dopamine cells. But people with Parkinson's disease actually suffer from a variety of non-motor symptoms: anxiety and depression, difficulty sleeping, constipation is one of the earliest symptoms, and some of these symptoms actually predate diagnosis. Now, they're not specific enough to be used to diagnose the disease any earlier, because people who are elderly suffer from constipation and sleeping problems, but we now know that these are organic symptoms of the disease. Again, not enough to be able to diagnose the disease with. So the idea is that there's a pretty good, physiologists have a pretty good knowledge of late stage Parkinson's disease. And that's sort of where I was trained with Chagai Bergman here in Jerusalem, and then with Charlie and Jim Surmise, in looking at sort of late stage Parkinson's disease. And we understand that as physiologists. And there's a lot of exciting work, again, looking at oscillations that Charlie had contributed to that occur in Parkinsonian brains in late stages. But most of the work on this prodromal period, where people realize, there's a pathology, a Parkinsonian pathology that happens during that period, most of that research is done by biochemists, geneticists, pathologists, and there isn't really much physiological work. And I think sort of the assumption in the field is that if there's this Parkinsonian pathology, alpha-synuclein, that's killing cells, and that's why it's killing cells in parts of the brain that are causing the symptoms. But that's that's the level of explanation. But really, physiologists haven't gone in and said, wait a minute, maybe cells aren't dying. And even if they are dying, clearly there's a period before they die where something is happening to neurons, and their physiological activity is obviously changing in response to this pathology. And we should be looking at that, because if we're able to look at the physiological changes, first of all, it can maybe give us a way to really have a better understanding of this disease process at the earlier stages, and possibly come with what I call physiological biomarkers. So if certain neurons are acting a little bit weird because they're beginning to have some pathology early on, and then that affects some physiological readout in the body, then maybe we'll have we could piece together and start putting together sort of these physiological biomarkers as indicators of the disease. So I think it's really, sorry, excuse me, I have this problem in my office where the light goes off if you don't move. So I think, you know, to complement this really good understanding of late-stage Parkinsonism, where there's a lot of understanding of the physiology, we need to be doing that at the earlier stages. That'll enable us to you know, maybe diagnose the disease earlier, but also maybe possibly come up with interventions, ideas for interventions at the earlier stages.
So tell us about why you're looking in the, so you're looking at sort of the sort of vagal targets that maybe produce some of these prodromal symptoms that you described, like constipation. So talk about can you talk about what we know? Those are, we see early evidence that there's Lewy pathology in those areas. Is that right? And yeah, can you just say something about where you're looking and how you're doing it?
Right, so we know, I mentioned the alpha-synuclein. We know about that pathology for over 100 years from the work of Dr. Lewy, and the pathology is named after him, but it's been sort of characterized more closely about 2 decades ago, that there seems to be this progression of pathology throughout the brain, and it begins in the brain stem. So there are regions of the brain that first exhibit these pathologies, and then it sort of expands. And there's a whole discussion about how that happens. Is it, you know, is it more of a prion-like disease? Is it not? What are the determinants? But so the idea is that one of the earliest places where that happens, and what the scientist, you know, Heiko Brack, would describe this, called stage 1 Parkinson's disease, which is a stage which is, you know, well ahead of diagnosis, happens. You find this pathology in vagal motor neurons, and motor neurons that innervate the GI tract, and they're located in the brain stem. And that's one of the first stations. So my idea is, let's, you know, if we model something like a Lewy body, and only in that position, we'll be modeling a very early component. We'll have a relatively, you know, well-defined pathology because it's only in one place. We put it there, and then see how that affects the animal, how it affects the phenotype, and then try to understand. And we found out that that is sufficient to cause this sort of Lewy-like pathology in that region, is sufficient to create a symptom in the animal models. In the mice, that's similar to the human constipation, which again we know is one of the earliest, if not the earliest, prodromal symptoms. So that sort of gives us a handle on a very early time point, and look at a, you know, situation where the cells, we can see that they're not necessarily dead, but there's definitely, the physiology changes in a dramatic way that then can be associated with the phenotype. And I think that, you know, we've worked on the vagus for quite some time, but I think that concept can be carried on to other symptoms. So, for instance, I'm involved with a bunch of colleagues in Europe on a project looking at anxiety and depression, and we're sort of doing the same kind of, looking at the same kind of questions, looking at the Raphe nucleus. We know the Raphe nucleus is related to a lot of things, but it's also been implicated in anxiety and depression. We also know that Parkinson's patients have organic anxiety. They're not depressed and anxious because they know they're sick, but this is actually a symptom that's organic. And it turns out that there are even dopamine cells in the dorsal Raphe nucleus, and it turns out that they may be selectively lost in Parkinson's patients. So Glenda Halliday has an old paper showing that there's loss of dopamine cells in that region, and that again suggests maybe we have the same kind of thing. We have, you know, loss of dopamine cells. We know that we have Lewy pathology in that region. Maybe that's messing up the physiology of you know, serotonergic neurons or dopaminergic neurons in the Raphe that are affecting the serotonergic neurons. And again, that you might be able to somehow look at the change in the physiology of those cells and connect it to a phenotype. So can we create an anxiety-like phenotype in mice by putting alpha-synuclein in that part of the brain? And the same thing, you know, I'm trying to work with a colleague about pain. We know, as I mentioned in my talk earlier, chronic pain is not very common, but it can be in some patients an early symptom. And again, we're looking at the involvement of areas like A11, part of the descending pain modulating system, which is also, you also find the Lewy pathologies in these regions. Maybe that's contributing to our level of pain sensation through the spinal cord. So I think it's a very rich paradigm and ought to be followed. It ought to, look at the physiological basis for these symptoms. And I think it'll give us a better understanding of this early time points of the disease.
So sorry, go ahead.
Oh, so I was just going to just ask about this idea of pacemaking, using some of the intuitions that we have from midbrain dopamine. There's a sense that there are ideas about the stressful pacemaker theory and how metabolic mitochondrial stress is actually causing cell death. And how well are those ideas? Is that a starting point for you in all of your exploration, regardless of where you go? And how well is that mapping on to for example, the dorsal motor neuron of the vagus?
Right. So no, that's definitely another aspect of it. Is this selective vulnerability? So why are certain cells affected more in one disease than others? And this is sort of what's the expression of the $1 million question in neurodegeneration? Because it seems like every neurodegenerative disease happens to have a cell type that's selectively vulnerable, and being trained by people like Jim Surmeyer, the thinking is, again, this notion that physiology of the cells, the pathophysiology of the cells might have a contribution to that. So why is there selective vulnerability? Jim Surmeyer showed that dopamine cells of one flavor in the SNC have a specific phenotype of calcium oscillations that causes them to have more basal oxidative stress, whereas the VTA, other dopamine neurons, don't have that, and they don't seem to have basal oxidative stress. And we know from human pathology that those cells are the ones that are selectively lost are the ones that are more vulnerable. So I'd worked in Jim's lab, and we kind of saw the same thing in vagal motor neurons, that they have this basal oxidative stress. But the interesting twist on the story was that when I got my own lab here in Jerusalem, I said, okay, so we know this is basal oxidative stress. Let's add on this additional stressor, this additional alpha-synuclein. Does that make it worse? And we expected it to make it worse because that's sort of what people had seen in dopamine cells. They saw that alpha-synuclein is synonymous to excessive oxidative stress. And in fact, we found that the opposite is true. Somehow, the vagal motor neurons, actually, when you add the stress, you either use a transgenic animal that has more alpha-synuclein, or you acutely inject it with alpha-synuclein, they're able to reduce their basal oxidative stress. They have some sort of adaptive response to lower the basal oxidative stress. That's really interesting. We were able even to show that in a specific animal, there was a divergent oxidative phenotype between dopamine cells in the substantia nigra and the DMV neurons. So they differ. The substantia nigra neurons, are worse off with the alpha-synuclein. The DMV neurons, somehow it makes them better off. And we saw several physiological indications of that. So that, again, makes this question of selective vulnerability even more interesting, because now maybe we're able to explain this trajectory. I mean, we know clearly that the most vulnerable cells in Parkinson's are dopamine cells, substantia nigra, locus coeruleus neurons. DMV neurons are lost in some patients, but there are many patients where there doesn't seem to be that much cell loss. So perhaps this ability to respond, maybe these vagal motor neurons have some trick up their sleeve that enables them to respond to this additional stressor by reducing the levels of oxidative stress. In a sense, it becomes protective. So we don't. That's what we see. We don't understand it, but it's a very interesting lead to follow.
So one theme in all of this is that the disease symptoms are usually caused not by the loss of some neuron, but by the reaction of some other neuron that isn't lost. It's now behaving badly. So the dopamine cells go away, it's true, but the main effect of that is that other neurons in basal ganglia start to fire in the wrong pattern. In the dorsal motor nucleus of the vagus, the neurons that are affected by the synuclein don't die, but they actually fire in the wrong way themselves, is without any intervening other kind of cell. Hey, Josh, your camera adapts really well to the.
We can see you.
Yeah, it's like there's a little flash, and then it's fine, but sort of you'd be sitting in the dark. Anyway, so what is the? It seems like it's a little bit simpler to think about the dorsal motor nucleus of the vagus neurons because there's not a second stage. It's not like losing them causes some other neuron to go crazy. It's more like the disease just affects them directly and changes their firing pattern. So what is it that changes about the firing pattern?
Right. So I mean, I think that's an important point, and in a sense maybe makes our work a little bit easier. I mean, we can't tie it clearly to Parkinson's disease like we can do with the end stage models and look at the basal ganglia physiology. But on the other hand, the brainstem is much closer to the effectors. So if you have some change in the brainstem, you could maybe have a clearer readout in somatic readout in our case in the GI tract. So that's a benefit because we can make a clearer connection between what we see happening to the cell and what happens in the body and the internal organs. And that's something that's been really difficult in Parkinson's. I mean, we see people have tremor and people have oscillations of the basal ganglia, and we want to believe that one is related to the other, but we can't really find a very simple association. The frequencies aren't even the same. The oscillations in the basal ganglia are 15, 30 hertz, but the tremor is about 5 hertz. So that in a sense it's easy. The prospect of being able to manipulate or perturb using these models of Parkinsonism, one of these brainstem areas, and then getting an immediate readout makes it easier. So in that sense, it's true. I mean, it's easier to correlate the physiological changes. What we've seen in vagal motor neurons is that they simply slow down, and vagal motor neurons are part of the parasympathetic system, which is the system that's supposed to the rest and digest component of our autonomic nervous system. So if the vagal motor neurons are slowing down, that means there's less parasympathetic tone, and that means that parasympathetic tone increases motility. Then if we have less tone, that can possibly explain reduced motility. And we know that constipation will be related to reduced motility. It's not that simple because most constipation has to do with the large intestine, which isn't really innervated that much by the vagal motor neuron. But at least it seems to make sense, and at least we're able to show the connection in the animal models. That definitely, if we make the vagal motor neuron slow down, then that directly affects the motility. So
do they normally carry some signal, or are they just sitting there going at some rate all the time? Or do they sometimes speed up, slow down, and that changes motility in the small intestine?
For sure, the parts of reflexes. So the vagal motor neurons, let's say looking at the cardiovascular system of the heart, so actually the parasympathetic tone there actually slows down heart rate. So we know that we have many reflexes that are there to maintain our heart rate and our blood pressure, and these circuits in that part of the brain are involved in that, in these reflexes. In terms of heart rate, it's actually another population of vagal motor neurons that come from another region of the brain, which maybe responds faster. So there's definitely our autonomic reflex components of our behavior are important with respect to these neurons, and that's what they're part of. And again, people with Parkinson's disease have orthostatic hypertension, so they have a lot. It's called general dysautonomia. Constipation is only one component. There's a general dysautonomia. The autonomous nervous system is off kilter, and a lot of it is the parasympathetic component. So again, we think that this can explain it. I mean, these circuits are pretty complicated. They're not as complicated as the basal ganglia, but there's still something to learn. I mean, about these reflexes, and that's actually something we might look at eventually. Is here in our study up to now, we've seen, like as Charlie you mentioned, a reduction in the basal firing. That's all we study. But there, of course, there's going to be a moment by moment component, and that could be interesting to correlate. Can we actually see the change in the vagal system corresponding to some change in the GI tract or in breathing or in heart rate? But we're not there yet.
So reflex means something special in this circumstance. I mean, most people's idea about a reflex, it maybe tracks back to the stretch reflex, where you sort of imagine that the motor neuron is sitting there doing nothing, and some sensory input comes in, and the motor neuron fires an action potential or two, and then some muscle moves. But these are not waiting for anything. They fire along all the time. So they function more like kind of like an analog signal, I guess, and their action potentials get integrated in the intestine to produce a relatively slowly changing signal that has to do with firing rate. Is that right?
Yeah, I mean, I think that's true. And we know, for instance, that our heart rate is under constant vagal tone. So our pacemaker, our sinoatrial node pacemaker, is much faster than it is when our heart is beating and innervated. So we're all under constant vagal tone. If we disconnect our heart from vagal firing, our heart rate would probably go up to around 100 beats per minute, but it isn't. It's usually around 70, 75, 60, and that's because there's definitely a vagal tone both on the intestine and on other internal organs, and yeah, in that sense, it does change. You also see changes in their firing rate with breathing. So all these components are sort of combined. You can look at the firing rate of these neurons and see them modulated by heart rate, modulated by breathing.
So I guess if they're firing at the wrong rate, the reflex circuit is effectively, looks like its gain is too low, or it looks like there's a signal there that isn't really there, because the actual firing rate of the neurons is the signal from that nucleus. And so basically the nucleus is constantly sending a signal that would normally happen, but would happen in some special time, maybe when digestion is supposed to be postponed. And so basically it's saying postpone digestion all the time. I mean, is this too simple of a way to think about it?
No, I mean, there's clearly huge literature and this description of vagal innervation is a little bit simplified because it might do one thing to the muscle, and the intestine has their own pacemakers. I mean, the intestine is also autonomous, so there are really interesting interactions.
These are interacting oscillators.
Right, they're interacting.
And the heart is also oscillator. So basically, this is a kind of neuronal connection between interacting oscillators that we're not really very familiar with. I mean, as neurophysiologists, it's not usually what we study.
Right. I mean, I definitely ought to be more familiar. There's, definitely previous generations. To this day, there are excellent physiologists who study this, know a lot about these, pacemakers and their interaction. And but I think it's time, to be able to understand Parkinson's disease, it requires us to really, put our money where our mouth is, and also begin understanding those interactions, the interface of the neuronal oscillators with intrinsic oscillators of the gut and the heart, etc.
Isn't it remarkable? Because in the basal ganglia, where Parkinson's disease other symptoms form, again, you have this group of interacting oscillators that are representing signals in a way that isn't the usual way of talking about the cerebral cortex or the spinal cord motor neuron, which is our most familiar neurons that we think about. And that has impeded our understanding of Parkinson's disease, because we couldn't really wrap our minds around interacting oscillators as a neuronal form. And there it is. I mean, a completely different system, kind of unrelated system. But then again, we see this problem of interacting oscillators being at the source of the problem. Coincidence? I think not. So how about anosmia? Another important prodromic symptom of Parkinson's disease. Maybe that one is a little better diagnostically because anosmia isn't super common among everybody.
Yeah, that's definitely true. And there are people who are working on it. There are definitely a lot of, I have somebody I know in Sweden I've recently met. I mean, there definitely are people looking at that, but that's more related to the fact that we also know that the Lewy pathologies actually affect, your olfactory system. It's also one of the early, I didn't mention it, but it's one of the earliest stages. And in fact, some people think that that's, there might be, that actually maybe where Parkinson's starts, and then it goes, somehow into the gut, and then from the gut back into the brainstem. So, but we definitely know that Lewy pathologies in the olfactory system are also an early stage of Parkinson's disease. So that's not something I specifically work on.
What is the effector that gets us from alpha-synuclein overexpression to breaking a pacemaker, or changing a KV4 density? Like, what do we imagine is the actual connection there?
So, I mean, over the years, I've looked at all sorts of hypotheses, and I think, maybe all of them might be correct, or some of them. One I used to think about was, we know that alpha-synuclein binds phospholipids preferentially. And I think it's very likely that one of the things that happens, although I haven't been able to demonstrate this, is that, for instance, we know PIP2 modulates calcium channels. So it could be that somehow, alpha-synuclein, too much of it is screwing up, is, maybe binding too much to PIP2, and then there's not enough of it to, so it's causing some effective modulation of calcium channels. That could be one thing that's happening. One thing we tried to propose in a recent study is that alpha-synuclein causes changes in neuronal morphology. And so in our case, we saw neurons shrinking, and we think that shrinking, if a cell shrinks, but it expresses the same amount of ion channels, then the current density will be different. And the thing that affects the integration and the changes in membrane voltage of a neuron is the current density, not the total current. So a cell shrinking, it's actually pretty natural that it can mess up. If it's not compensating for the amount of channels it's expressing, it should change the way it voltage charges. And if it's a pacemaker, it's going to change the pacemaking. So that's one thing we believe we have demonstrated, but maybe we could do some more work on that. So yeah, there might be all sorts of other interactions that alpha-synuclein. This also requires, a better knowledge of cell biology, which is really not my forte.
But whatever it is, you imagine that it would be something that sort of propagates transsynaptically, right? Because the idea is that this is an expanding syndrome, right? Or is that a kind of a toy idea, or there is actual data that it expands?
Definitely, the Brock hypothesis is that that's what happens, is that somehow there's transsynaptic, movement of alpha-synuclein that then, nucleates in the next station and goes on. And that's why some people like to call it a prion-like disease. And definitely there have been a lot of animal models that have shown this, that have shown that this is possible. So if you put alpha-synuclein in one part of the brain, you'll end up finding it somewhere else. Or if even in humans, if it was I think in a graft, if you put a graft and the graft is healthy, eventually it'll develop the alpha-synuclein. So we know that's possible, that happens. But on the other hand, it really isn't a prion disease in the sense that it goes from every neuron to every neuron. There are certain targets, and it doesn't even seem to expand exactly according to the connectome. So So that's only one component. The connectome is one component, which cells are connected to the other. But then there's something about a cell autonomous component, about specific neurons that seem to take it up more, affect it more. And I think that's, that's part of the story. So, we're interested in that aspect. It's okay, it seeds in a certain cell type. How does it change that cell type phenotype? And we're sort of agnostic about, actually, our project, the project that I talked about, an hour ago, is sort of agnostic about this question of whether alpha-synuclein is moving around, because we're just saying, okay, we have it in one point. Here, it's already doing something interesting, irrespective of the question of whether it's moving between cells or not.
So we think of all the basal ganglia dysfunction as rooted, at least I from my simplistic understanding, that it's rooted in this loss of dopamine, and that everything else is sort of this compensation that happens. But is there reason to think that there are cell autonomous things happening in the other rhythmic or the other oscillators in the basal ganglia? Or, I mean, are there thoughts about that?
So, I mean, of course, that's something that, Charlie has worked on extensively, and, other people, that we know that once there's a loss of dopamine, then other oscillators that are not, neuromodulators in the basal ganglia, but are actually the main players, the basal ganglia, their firing gets screwed up. In fact,
but do they have Lewy? I mean, is that secondary to Lewy pathology, or this, whatever the cell autonomous part of this is, and not the network effect?
Actually, the striatum, which is the main, part of the basal ganglia, there really isn't a lot of alpha-synuclein pathology in the striatum. So really, that late stage of Parkinsonism really has to do with loss of dopamine, and then maybe changes of gain in the circuit, and all sorts of neurons responding to the loss of dopamine directly, or loss of secondary effects of what loss of dopamine does. The alpha-synuclein pathology is less important specifically within the basal ganglia. It's more important, I think, earlier on. But it definitely contributes, very likely contributes, or is associated with, very highly correlated with the loss of dopamine cells. So once the dopamine cells are gone, that starts a whole new can of worms, which is sort of being studied for many decades already. But again, I'm more interested in the regions that are directly affected by alpha-synuclein, and how those cells respond, and how they may lead to certain prodromal symptoms.
So I was wondering if the synuclein pathology is controlling motility in the small intestine, but not the large intestine. But most constipation, you say, is caused by changes in the large intestine. Then is there a way to directly measure small intestine motility as a possible diagnostic tool?
Right. So that's that. So I actually met up with a gastroenterologist in the hospital nearby, who has one of these smart pills that you can swallow something, and sometimes they have a camera, but they actually don't have to. They can measure things like temperature and acidity, and then they know, depending, what part of the GI tract, because every part, the stomach is different from the small intestine. With you look at all those parameters, if you measure them, you actually know when it moves from one to the other, and then you can measure in a human how long time of passage. So in principle, that's one direct way of looking at it. And again, if we become convinced that what we discovered in an animal model is true in humans, that could be a diagnostic. Another aspect is, again, vagal motor neurons innervate the heart, so you could also look at changes in heart rate. I mean, people studying Parkinson's disease have thought about these things. There have been studies about heart rate variability in Parkinson's. They haven't been very conclusive or very helpful. But we think that if we're right about this sort of vagal neuropathy, that this reduction in vagal tone occurring because of alpha-synuclein accumulation in the brainstem, then maybe there are specific challenges like atropine, which is something that stops the activity of the parasympathetic system. So maybe you could maybe put somebody up for a test where you look at their heart rate recovery after exercise, and look at how you challenge that with atropine, and see what is there a change in the vagal component. So I mean, I'm not a clinician, I'm not a gastroenterologist, I'm not a Parkinson's clinician, but I'm trying to speak to people like that and convince them to maybe try things like that on groups of patients to see whether any of that is viable. Any of those ideas are viable. I don't think there's any clinician who would be convinced right just now, but maybe over time, if we can demonstrate that this is a real thing that happens in patients, it might catch on more.
What's the advantage of early diagnosis of Parkinson's disease? Is there a particular advantage to knowing?
So yeah, everybody asks me that. It's a great question. I think for a specific person with Parkinson's or a person that's worried about developing Parkinson's, maybe some people might be interested in knowing earlier, but a lot of people might say, I don't want to know a day earlier than I need to. Why be anxious about it? Why? But I think as a field, we want to be able to, move to an earlier time point. And the reason being that, as you know, for Alzheimer's disease and Parkinson's disease, most of the therapies that have been tried and tested, spending billions of dollars over the past 20 years have failed. Colossally, one after the other, nothing works. Including a recent trial that was sort of grew out of the Surmeyer calcium hypothesis. And of course, one of the reasons these things have failed is it could be that even though the biology behind it makes sense and it's been demonstrated in enough models, but you might be trying it on humans who are way too late in the game. And if we were able to find an earlier time point and say, okay, now based on anosmia and your vagal output and something else and anxiety, we think you have a high likelihood of having Parkinson's disease. Maybe some of these therapies that have failed today, you could try them again at an earlier time point and suddenly you would see that they work. I don't know if we can convince, once a clinical trial fails, I don't know if you can convince pharma to try it again, but at least it makes sense. If you have an earlier time point, maybe these therapies actually do work.
Most ideas about Parkinson's disease are neuroprotective. How can we protect the dopamine cells? And I guess maybe we don't have a perfect neuroprotective strategy for dopamine cells, but when we do have one, it's going to be too late if we give it to patients who've already lost all their dopamine cells. So I think, don't we need to be preparing for that time by figuring out what the time sequence is from the first detectable thing? And that's what I think about it.
Of course, I mean, I'm putting this emphasis on physiological biomarkers, but of course, there's a huge industry and academia, it's working on, real biomarker, what other people consider, most scientists consider biomarkers, I don't know, sebum, the stuff you exude from your skin, all sorts of other, possible indications of alpha-synuclein. In fact, I think the nerves that innervate our sebaceous glands actually exhibit alpha-synuclein accumulation. So there are some biomarkers that people are taking skin punches to try to discover. So I was listening to and I was at a conference at the Van Andel Institute in Grand Rapids a few months ago, and there's a lot of optimism that within 20 years from now there will be various validated and reliable biomarkers for earlier diagnosis of Parkinson's disease. Hopefully, maybe the physiological one will be one of them, but there are also definitely biochemical ones, and so I think there's reason for optimism for moving towards earlier diagnosis for Parkinson's disease in the next 20 years.
I'm wondering if understanding some of these these physiological markers in these early tissues could serve as like an organizational principle for understanding homeostatic mechanisms. Like we heard a talk about proteostasis last week, and this idea that there are these programs that kick in, that if you just sort of understand what those little networks are that get triggered, with some kind of insult, like this, presumably the alpha-synuclein overexpression, that those can somehow be triggered differentially, or different types of modules could be triggered in different cell types, and that could somehow give us a sense of where we could intervene, and at which point, if this is an expanding syndrome, like how do we arrest it, like at one point, right? I mean, it seems like there would be clues there, right, in understanding how cells respond to this type of overexpression, if we imagine that that's how it works.
Right, so I mean, homeostasis, of course, there's a network component, which is, just as Charlie mentioned earlier, that there's this divide between the cortex and cortex-oriented people thinking about neurons as not as pacemakers, but being driven by input, and people who study pacemakers where they actually can drive themselves. So the same thing, the questions about homeostasis in cortical networks usually focus on network homeostasis. People like Gino Torrigiano, and that's a huge area of research. But again,
I'm talking about cellular mechanisms, yeah.
Right. So cells that are pacemakers, I think really the emphasis is on this cellular homeostasis of maintaining a certain range of firing rates, and I'm certain that those things exist. So we think, for instance, in the context of the DMV, that there's that at least from our data, and it's not I wouldn't say we've proven it completely, but there's an indication that these homeostatic mechanisms are at play to a certain extent, because some currents, some channels seem to be homeostatically regulated. So we think that we see calcium channels being homeostatically regulated, but on the other hand, others aren't. So there's a screw up, and that's what basically throws the cell off. On the other hand, it could also be actually protected. Maybe what we think is not working properly is actually what the cell is intending to do. It wants to have a larger potassium current, so it wants to slow down, because if it slows down, that means less calcium influx. Less calcium influx means less oxidative stress that's driven by calcium influx. So yeah, I mean, I think definitely, thinking about homeostatic responses is really important. I mean, as Charlie mentioned earlier, a lot of the symptoms of Parkinson's are not necessarily the initial effect, but rather, the maladaptive homeostatic response that turns out to be maladaptive in certain respects. So yeah, I mean, I think it's definitely an important aspect of thinking about these issues.
There's two kinds of homeostasis. I think a lot of cell biologists think homeostasis is a device used by the cell to take care of itself, to make sure it doesn't die, that kind of stuff. But homeostasis could also be the cell trying to make sure that it continues to perform its function that's required by the rest of the circuit. And those might be in conflict, just like happens with people. And so the homeostasis that you need from these neurons is for them to keep their firing rate and to keep their responsiveness to inputs, even if maybe it puts a stress on them, their mitochondrial enzymes and stuff. Let's hope that all the cells think about their responsibilities to the brain rather than simply their own survival.
Yeah.
Are we gonna end on that note, Josh? Do you want to have the last word rather than that Borg-like?
I mentioned that I'm an alumnus of this lab, so of course for me it's a real honor, and it's to be back and be able to speak to Charlie again, talk about these things, and we still have points of interest, and I hope to visit in person after this pandemic, and we can pick up and continue talking about these ideas in the future.
Definitely looking forward to that.
Hope to see you soon. All right, thanks guys. This has been Neuroscientists Talk Shop. Thank you, Josh Goldberg. Thank you, Charlie. See you guys next term. Bye.